It's so lovely to be here with you all here at this beautiful facility here. I've never been up to Mammoth before, and I've really enjoyed spending a couple of days here. And so thank you all so much for coming and for that really nice introduction. Carol already said this, but tonight I'll be talking to you about my research and the research of some of my colleagues. And we look at reconstructing Earth's past temperatures to understand future climate change. So as she said, I'm Dr. Jamie Lucarelli. I started my educational journey in community college, just to tell you guys a little bit about me. So I started at Los Angeles City College. And prior to that, I actually was a hairstylist. So I wasn't always a scientist. I was a hairstylist that loved science and decided to go back. And then I transferred to the university at UCLA, and I got a bachelor's degree in chemistry, a master's in geochemistry, and then a PhD in geochemistry. And so currently, I am a postdoctoral fellow in the Tripathi Lab Climate and Biogeochemistry group at UCLA and part-time faculty at Cal State San Bernardino. I live up in the San Bernardino Mountains in a little town called Crestline. You can see a picture there. That's a picture of Crestline. It's not really too different from here. It's just more densely forested, I would say, but has lakes and it's high elevation. And things that I love when I'm not doing science is my beautiful family, my kids, and I love to watch Harry Potter movies. I love my dog. He's a little dachshund piggy, and I love a good meme. So I put a nice little science meme for you guys there. Tonight in the talk, I will be starting out with some background information that is relevant to climate science, both modern and paleoclimate, just kind of a primer on some of the topics we'll discuss. And then go into the basics of the method that I use, which is called carbonate clumped isotopes, and talk about what that is. Go into how we collect samples and how they're processed, and then how we take data using mass spectrometry, and then go into some results and conclusions that have been determined using this method. So starting with the background information. So probably a lot of people here know this, but just in case, what is a greenhouse gas? So specifically, these are gases in our atmosphere that trap heat. So not all gases are greenhouse gases, only ones that absorb infrared radiation. And how that happens, you can see in the top figure there, is you have energy coming from the sun. It radiates down to Earth, and then some of it is reflected back as infrared radiation, which you can see if you look at a street on a hot day, like a black street, and you can see the wavy lines coming up, and that's that heat being reflected back. And that is actually absorbed by some gases such as carbon dioxide, which is CO2, carbon with 2 oxygens, methane, which is CH4, so a carbon with 4 hydrogens, and then water vapor, just H2O gas. And then moving on to what a climate feedback is. So that's something where when you change one quantity, you actually end up changing a second quantity. A very relevant climate feedback is something called albedo, and that's how reflective something is. So looking at the second figure there, you can see snow and ice, which has a very high albedo or high reflectivity. So if you have energy coming in from the sun, about 90% of that is reflected back out to space. So that helps keep the Earth cool. But if you start melting ice, then you expose the darker colored water underneath, and that has a lower albedo or lower reflectivity. So you're only reflecting back about 6% of the energy coming from the sun. So that can cause warming. And then the feedback comes in where you're warming, and that causes the ice to melt, and that causes more warming, and then it kind of just goes around in a circle as a feedback. So what is paleoclimate research? That's the type of research that a lot of people, myself included, in our group does at UCLA. So what does evidence reveal regarding how major events in geologic time affected Earth's climate? So we try and find different records going back through geologic time that can inform us how our climate has changed through things like volcanism, tectonic uplift, and things like Milankovitch cycles, which are the way that our orbit changes around the sun over long timescales. Other things that we try and investigate are what are the potential consequences of greenhouse gas emissions based on past evidence. So in the past, we can look at specific time periods that are believed to be warmer and had higher concentrations of carbon dioxide or similar concentrations to today, and try and understand how that affected temperature, how it affected things like permafrost, which is what you see in that top picture there, and ice coverage around our Earth in places like Antarctica. Some methods that are used for paleoclimate reconstructions include things like ice cores. So this is a picture of NASA scientists that they're at Lake Vostok in Antarctica, which is an extremely cold area, which is close to the interior of Antarctica. And they are taking a large drill and drilling down as deep as they can into the ice to access very, very old ice that is believed to be up to 400,000 years old. And the reason for doing this is that tiny little bubbles of gas actually get trapped within the ice, and you can extract that gas and take measurements of it to understand how things like carbon dioxide concentrations, the greenhouse gas, has changed through time, and how that correlates to the way temperature has changed through time. So that's what we're looking at here in this plot, where we're looking at the temperature at Lake Vostok, and then also on the right, the CO2 concentration. So you can see that they're pretty tightly correlated going back up to over 300,000 years ago. And then on the far right, you can see that big spike in 2016, and we're even beyond that concentration now. In 2016, the CO2 concentration was about 400 parts per million, and currently it's about 420 parts per million. So we're trying to understand, based on past evidence, what sort of temperature increase might we see. Other evidence can come from areas like dendrochronology, which is looking at tree rings. So as a lot of people know, trees get one ring per year, but you can get a lot more information because a tree ring will be closer together. If it's a time of drought, they'll be farther apart if it's a lot of precipitation in that particular year. And you'll see things like scarring from forest fires. So there's all sorts of information that you can gain from looking at tree rings. And they also can be carbon dated, so you can have really tight constraints on when these things happened going back through time. Another method you can use is actually looking at geologic formations, so rock formations. Down on the bottom there, you can see a really nice example of what's called folding. So if you were to observe that, you'd be like, okay, this particular area had a lot of tectonic activity during a particular time period. And then on the top there, maybe the formation has shells in it. So that could give you information about maybe this used to be a lake, or it used to be part of an ocean, and now maybe it's a desert. So you can get all sorts of information just by looking at rocks. And the last method here, there are others, but the last that I'm going to discuss is using sediment cores. So these are similar to ice cores, but instead they're taken from sediment. And so you can get all sorts of information from these, like looking at pollen or different plants that are present and how that's changed through time, and different shells that are present, and look at their geochemistry, which can often be related to temperature. And that brings me to the specific method that I use, which is called clumped isotopes. So what an isotope is they are atoms with the same number of protons in the nucleus, but different numbers of neutrons. So what that ends up meaning is that you can have atoms of the same element, but they weigh slightly different amounts due to these differences in their nucleus. So looking specifically at carbon, so these are the two carbon isotopes, and they have the different numbers of neutrons in their nucleus, and you can see highlighted in yellow there, the one on the far left weighs 12 daltons. So a dalton is just a unit of measurement. People are often weighed in pounds, and atoms are so tiny that they are often weighed in what's called daltons. So you have the mass 12 carbon isotope, and then the one right next to that is the slightly heavier isotope of carbon that weighs 13 daltons. And then looking at oxygen here on the right, the one on the left weighs 16 daltons, and then, or the one on the left weighs 16 daltons, the one on the right weighs 18 daltons. So those are two of the most common oxygen isotopes. These are very useful because they can actually occur together in the same molecule. So here you have a carbon-13 in the same molecule as an oxygen-18. So that's what's referred to as a clumped isotope. So you have two clumped together in the same molecule, and how many of these occur within a sample is temperature dependent. So this is what we're actually looking for. And we look for these in carbonate minerals because they contain both carbon and oxygen. And they also occur very frequently in nature. So in all sorts of natural environments, you take calcium, you add it to carbonate, and you get a carbonate mineral, something like calcite or aragonite. And here down at the bottom, this is a chemical equation showing what's happening right at the surface of formation of one of these carbonate minerals. So you have this isotope exchange reaction, and so this is the carbonate molecule, the one that's got the red square around it with the heavy carbon, the heavy oxygen in the same molecule, and it has a mass of 63. So the total mass, if you add 13 plus 18 plus 16 plus 16, this is your mass 63. Doubly heavy isotope substituted carbonate molecule. And these are directly related to temperature, their abundances. And it's really a great technique because carbonate minerals are extremely common in nature in so many different settings. So these are all different samples that we've used in our lab at UCLA. So starting at the top left there, that's a foraminifera. They're these very, very tiny shelled organisms that live throughout almost every ocean in the Earth. And they're most of them are about the size of a grain of sand, but they produce these really beautiful shells. The second one, coral, they make their skeletons typically out of aragonite, which is a carbonate mineral. Huge limestone formations, all sorts of shelled organisms like snails and clams. There on the top right, those are dinosaur eggshells, which also contain carbonate minerals. Then you have a megalodon tooth. Then those are large cave carbonate mineral formations called speleothems. Then you have the little white specks there in the soil. Those are carbonate minerals that occur within soils. And then the final picture there on the bottom right, those are called stromatolites. Those are some of the oldest known biologically created minerals on Earth. They can be up to billions of years old, and they're also made out of carbonate minerals. So using all these different samples, we can examine how many of these clumped isotopes they have and determine what temperature they formed at. So what temperature maybe the ocean water was that your very old shell grew in, just as an example. So it's a really neat method. So moving on to the way that we collect samples, a pretty common way is using the ocean drilling program. So this is a really neat program that sends scientists out, such as this is one of my friends and colleagues. His name is Bryce Mitsunaga at Brown, and he's out right now on a scientific cruise vessel where they take their vessel around different areas of our oceans, and they use this large drill to extract cores from the bottom of the ocean. So they drop the drill down all the way through the water, take out these cores, and then these become available to scientists. So if you want to study oceans in a certain region, you can request a core from the ocean drilling program from that particular area. Within these cores, you can find things like foraminifera, clams. You can look at the soils, so there's all sorts of information that you can take from these ocean cores. You can also find field-collected samples. This is another friend and colleague of mine, Professor Victoria Patrician from USC. And she's out looking for carbonate mineral samples in the field. You can find things maybe like fossils or corals or dinosaur shells, different soils, or maybe you would like to look in a cave for some speleothems. And then once you've collected your sample, depending on its size, there's different ways that it needs to be processed. So this was a student from Santa Monica Community College that I worked with. Her name's Ishal Trujillo, and she's removing the sediment from our samples from a core. So it's surrounded by a lot of sand and sediment, and you want to remove as much of that as you can. And if you're looking for foraminifera, which is there, that's a picture from under a microscope, you want to put your sample under a microscope and actually take tweezers and painstakingly select the specific foraminifera that you want from these samples. If you have a larger sample, it needs to be drilled or crushed into a powder before we can analyze it. And if you have a field collected sample, oftentimes they are larger, something like a big piece of limestone. So that will need to be drilled using something like that little drill there, or a mortar and pestle, which you can see on the right. And you drill it and make it into a fine powder. And then the last step, no matter what sort of sample you have, is to weigh 0.5 milligram of your carbonate mineral into a silver capsule. So that's what that picture is there. And you use something like this balance that you see here to get a very precise amount of 0.5 milligram. And once you've done that, you're ready for your sample analysis using a mass spectrometer. So at this point, you take your silver capsule that has your crushed carbonate mineral inside of it, and you put it into the carousel, which is right here. And so once you put it in there, you're going to drop your carbonate mineral, which is a solid, into a bath of phosphoric acid. And that will dissolve your sample, and it will turn it into a gas. So what's actually happening, if you look here, you have your starting carbonate sample, so CO3, that's carbonate. So each molecule has the carbon and the 3 oxygens. And like I said before, we're looking specifically for this mass 63 carbonate molecule with the 2 heavy isotopes in it. So when you drop it into the phosphoric acid, you will actually remove one of the oxygens in order to turn it into carbon dioxide gas. So by doing that, you end up with mass 47 CO2 because you've removed this one oxygen. So your CO2, now that it's a gas, will travel through a series of cryogenic traps. They're very cold. They have dry ice and liquid nitrogen in them. And as your CO2 gas passes through them, that will freeze out any water contamination that might be in your sample. And then after that, it will pass through what's called poropack, which is a special type of polymer that will bind with any sort of organic contamination. Say that if you have something from the ocean, it might have some organic contamination that needs to be removed. And once your sample of CO2 has been fully purified, it will enter over here where it becomes charged, and then it will go around a magnet here, where the lighter molecules are more strongly affected by the magnet and more they're bent around this curve on the inside path. And then the heavier molecules are less affected by the magnet, so they go around the wider path right here. And then these different masses are detected in an ion detector. And that will give us our information about how many molecules that we have with a mass of 47, which is exactly what we're looking for. So once we've done that, our mass 47 abundance is termed delta 47. So that's what you see right here. That's our final thing that we get from our computer, and after all the corrections are done, we get our delta 47 value. And what we do from there is we can use a relationship like this, where we have our delta 47 value and then temperature in degrees Kelvin, which is just another temperature measurement similar to Fahrenheit or Celsius. So for example, if you measured your sample and you got a delta 47 value of about 0.6, you'd be like, okay, so I know that my sample was formed at 30 degrees Celsius, which is the temperature right here on the line. So that's what these numbers are, is temperatures in degrees Celsius. So this is just an example of how we use our data to understand the temperature that our samples were formed at. All sorts of studies have been done since about 2006 using this method. So it's actually a pretty new method, and that's because these types of molecules are actually in extremely low abundance, and so it was only until about 2006 that they could even be measured. So that was when the first measurements were done. And since then, this method has really taken off, and now there's groups all around the globe that use this method to reconstruct past climates. So some examples are reconstructing ocean temperatures, the way that temperature has changed from tectonic uplift, and this has even been used to reconstruct the body temperature of sauropod dinosaurs, which I'm going to describe because everyone always loves talking about that one. So the first piece of research that I'm going to talk about looked at some samples from the Pliocene, which is about between 4 and 1 million years ago. That's what MYA stands for, is million years ago. And their samples were from Tibet, from an area that has a lot of permafrost, which is permanently frozen ground in high latitudes. And so the interesting thing about permafrost is it acts kind of like a lid, and it locks in a lot of frozen organic matter. And once this melts, microbes will start breaking down that organic matter, and that releases a lot of greenhouse gases like carbon dioxide and methane, and that can go into one of these feedbacks that we talked about before. So you get temperature that's warming, then you start melting your permafrost, which emits greenhouse gases, which furthers warming, et cetera, and that cycle goes around. And so time periods where this type of melting may have been occurring has been a target for paleoclimate to try and understand how this affected temperature. This research was performed by Professor Feng Chang from the University of Rochester and Professor Aradhana Tripathi from UCLA and Alexander Arnold, who's a graduate student at UCLA. This was a picture of their samples here. So these are the Pliocene to Pleistocene aged soil carbonates from the Tibetan Plateau. This was the region right here of the permafrost in Tibet. The conclusions that they drew from these measurements was that there was a very warm period between about 3 and 4 million years ago. You can see right here on this figure, where you have your temperature. This SLST is the summer lake surface temperature from a lake here on the Tibetan Plateau. And then this is the time going back from about 1 to 4 million years ago. So they determined that this time period here was quite a bit warmer, to the point of where the permafrost would have completely melted. And this was called the mid-Pliocene warm period. That's what this means. And then after this mid-Pliocene warm period, there was cooling of about 8.1 degrees Celsius. And then at this point, you had more glaciation and the formation of permafrost that would occur. So the interesting thing also about this particular time period is that the temperature during the mid-Pliocene warm period was believed to be only about 1.2 degrees warmer than we currently are right now. So this type of permafrost melting might be occurring in our very near future. It's already being observed now. And so we might end up at a point where we have a lot of permafrost melting similar to what occurred during this time period. The next piece of research I wanted to talk about goes back even further in time, looking at the Eocene between about 56 and 34 million years ago. And during this particular time period, it was believed to have what's called a greenhouse climate. And that means that they had a lot of greenhouse gases in the atmosphere. So this particular time was believed to have CO2 in parts per million of 560, which is over 100 more parts per million than we have at the moment, because right now it's about 420 parts per million. And what they wanted to understand is that during this very warm greenhouse climate, how did this affect what's called the latitudinal temperature gradient? So if you look here at this map, this is the way that our temperature changes across the Earth going through different latitudes. So here at the equator is 0, and then starting at about 66, that's the polar latitudes. So right now we have a pretty big temperature gradient from the equator to the poles. But as the Earth warms, the poles tend to warm faster than the equator does due to what's called polar amplification. And that means that the polar temperatures are warmed faster because of all of the melting of sea ice. And you can see that this is a plot of current temperatures. So here you're looking at temperature change, and the time on this axis is 1950 to 2020. So the global temperature's risen about a little over 1 degree Celsius, but the Arctic has risen over 3 degrees Celsius. So that's an example of polar amplification. So this study aimed to look at how did polar amplification affect the climate during the Eocene greenhouse climate. This research was performed by Professor David Evans at Yale. The samples were Eocene age foraminifera, and they were taken from all of these locations, which you can see by the symbols there on that map. So they were taken from all those different areas so that the latitudinal temperature gradient could be examined. So the conclusions that they drew was that the temperatures were indeed warmer than they were today on the sea surface at the sea surface. So let's look at this figure. There's a lot to unpack here, so I'm going to go through it kind of piece by piece. You have temperature and then latitude. So -80 would be on the South Pole, and then +80 would be on the North Pole. Here, this red shaded area, that is a computer model simulation of what the computer determined the temperature would be if we had a CO2 concentration of 560 parts per million, as it was believed to be during this time. These filled symbols, these are the measured values. So these were the temperatures determined using clumped isotopes. And so as you can see, they are a bit different from the computer model. Here in the gray shading in the center, this is the sea surface temperature today, and the lighter gray area around the outside is the seasonal variation in our current sea surface temperatures. These horizontal lines are the temperatures from the Eocene of the bottom of the ocean, so the deep ocean temperatures. So some important things that you can take from this figure is that paleoclimate information can be used to make models more accurate. This particular model, it made it look like there was still a pretty large latitudinal temperature gradient, whereas the measured values were saying no, these were actually pretty flat, and most of the temperatures were similar from high to low latitude. So this can help us improve our computer models by actually using data to inform our models. And also, it is worth noting how much warmer the sea surface temperature was during this time. And so that it could be that in, say, another 100 years, this could be the temperature of our oceans, because at that point, our CO2 concentrations will be very similar to what they were during the Eocene. And likely it will actually probably occur before that. So moving on to the kind of fun application of the dinosaur body temperatures. This research was performed by Professor Rob Eagle at UCLA, who is a collaborator of mine. And so the samples that he used, the first set, were what are called oviraptor eggshells. So you can see the picture there, an artist's depiction of what an oviraptor may have looked like. So these are non-avian dinosaurs, meaning that they are not direct descendants of birds, but they were very bird-like. They had beaks and they had feathers. And so they were wondering how similar were oviraptor body temperatures to, say, a reptile or a modern bird. Which were they more similar to? So that is the data for the oviraptor eggshells, and it's compared here in the green symbols to modern ectotherm eggshells, so those are reptile eggshells, and then in pink you have modern bird eggshells. So they ended up discovering that they were pretty similar to the reptile temperatures, but they did seem to elevate their body temperature maybe just a little bit. So maybe they weren't quite an endotherm, which means an organism like us that does fully regulate our body temperature, but they weren't exactly like a reptile either. So maybe they were somewhere in between. And then the next set of samples that they used were from Camarasaurus and Giraffatitan teeth and Titanosaurid eggshells. So these were sauropod dinosaurs, which were a lot of people's kind of favorite dinosaurs with the really long necks. And you can see how large they were compared here to a modern elephant. And what they ended up determining for these samples is that they did likely regulate their body temperatures, which was a pretty interesting finding because previously it was not really known whether they were more like a reptile. But by actually using data to examine their body temperatures, they determined that they did likely elevate their body temperatures and were more similar to modern endotherms, meaning they regulated their temperature. And the last piece of research that I wanted to discuss with you is kind of a cutting-edge application of clumped isotopes. This is something that myself and my colleague, Dr. Zishan Parvez from UCLA, have been working on for about the past year. And so this does not have to do with reconstructing temperature. So this is a completely different application of clumped isotopes. And so the samples that we used, you can see here, they're actually from California, from an area called the Cedar Springs. And these springs are very interesting geologically and also very interesting for climate scientists, and I will tell you why. So these springs, they are surrounded by a peridotite rock formation. And when rain comes down, it absorbs carbon dioxide from the atmosphere. And when that interacts with peridotite rock, it forms spring waters that are extremely high pH. Most natural springs have a pH of somewhere between, say, 7 and 8.2. And these particular springs have a pH over 11.5. So they're very alkaline springs. And this very high pH water forms a lot of carbonate minerals, which is the type of samples that we use. You can see that here in the white, the different carbonate minerals that are forming here. And this is very interesting to climate scientists because as these springs form carbonate minerals, they do that by removing carbon dioxide from the atmosphere. So they're what's called a natural sink of carbon dioxide. So they're removing it just naturally. And so we were wondering, first of all, how fast is the CO2 really being removed by these natural springs, not just in California, but there's an even larger formation of peridotite in Oman, which is in the Middle East. And so we wanted to understand how quickly the CO2 could be drawn down by the formation of these minerals, both naturally, and then also if you were to fracture this rock and introduce a feed of CO2, so this would be an enhancement of the rate, how quickly could you remove CO2 then? So what we did is we used our clumped isotope delta 47 value, which is related to the rate of mineral formation. So we used modeling and measurements to determine the rate that these minerals were forming at, and then we used those values to inform this right here on this plot, which is the amount of CO2 sequestered in tons. So that's the amount of CO2 removed. And then you have years. So going forward in geologic time, basically, how much would be removed naturally? And then if we were to artificially enhance this, how much could we remove? And we ended up calculating that we could actually remove the entire amount of CO2 that's ever been emitted in these rock formations. Now, actually doing that in practice would be really, really hard, and it would take lots of fracturing and drilling and all sorts of different things that probably we wouldn't want to do because it could damage habitats and things like that. But you may be able to do this on some sort of a scale and also use this to inform the development of technology to sequester CO2 in a completely environmentally friendly way, because these carbonate minerals are completely non-toxic and they also stay stable on geologic timescales, so they wouldn't re-emit the CO2 back out into the air. So this was our kind of new research that we just submitted for publication recently. So my take-home message for you that I hope that you get from my talk this evening is that clumped isotopes and other paleoclimate methods are very useful, and they can help us understand climate change that we may see in the future. They can help us inform climate models and make them better and more accurate. They can help us understand the way that our climate has changed in the past from all sorts of things like tectonic activity, volcanoes, and more. And they can help us understand things like how body temperatures have changed with evolution. And then there's all sorts of other exciting applications like how we can understand CO2 sequestration and mineral formation. So thank you guys so much. It's the end of my talk. Thank you. I'm happy to take any questions. Yes? They're not super common. There is a few large areas of them. We have a decent sized one here in California. The one in Oman is really, really large compared to the one here. I can't remember how many square kilometers it was, but it was like 10 times the size of the one here. And there are others, but I couldn't tell you exactly how many that there are.
Can that stuff be extracted and used in, say, power plant scenarios?
So there is some ideas kind of like that, where you use what are called mine tailings. So if you were, say, mining some sort of mineral and you end up with mine tailings that has the right properties that it could be used in the same sort of a way where if you introduce a feed, you'll form carbonate minerals. And people have tried this, and it actually does work, and it can be enhanced with things like microbes that will also at the same time draw down CO2. So that is some of the technology that's been proposed, actually. So, yeah.
And if you have questions, we'll just use this.
Oh, okay.
So that our Zoom audience can hear your questions as well. There we go.
All right. Thanks, Carol.
Yep.
My favorite question, why are the Milankovitch cycles asymmetric insofar as the warming is very rapid and the cooling is gradual?
I don't have a great answer for that. I haven't studied Milankovitch cycles in that much depth. I'm sorry. I know that at the moment, our Milankovitch, where we should be on the Milankovitch cycle should be a bit of a cooling trend. So instead, we're seeing warming, and I've been reading about that recently. But other than that, going back through time, I know that if you look at the plots and the periodic cycles of the Milankovitch cycles, and so there's actually kind of some questions about them, because at one point it was like every 100,000 years, and then it switched to about every 41,000 years with the way that it affected climate. And so I don't think all the questions have really been answered to my knowledge about Milankovitch cycles. So sorry I don't have a better answer for you for that.
Sorry if I missed this, but why do you hone in on the mass 63 number and delta 47?
Sure.
Versus the other variations?
Yeah, that's a great, okay, I don't know if I can pull the slides back up, but basically we use those particular molecules because they contain 2 heavy isotopes. So the other ones only either have no heavy isotopes or they have 1 heavy isotope. So in order to have this really nice temperature relationship, we need to have 2 heavy isotopes present in the same molecule. So that's why. And there are others, might I add, that have 2 heavy isotope substitutions, and they're even in lower abundance than the mass 63. And so there's actually some really great kind of even more cutting edge applications using the mass 64 carbonate molecule. And I spent actually a huge portion of my PhD developing that. As I didn't discuss that tonight, but that's another application. Yeah.
So you might have mentioned this and it went past me, but how is the curve of the carbonate 47 versus temperature, how is that curve derived?
Yeah, so the way that it's derived is by a couple different ways. So we can use things like precipitations that are done in the lab, and I've done a lot of these myself, where you produce calcite usually or aragonite under controlled temperatures. And so you get those, and then you run them in your mass spectrometer, and you're like, okay, these are the values that we get. Before that was ever even done, the delta 47 values were predicted with theory. So that was kind of the very first knowledge that this could even work, was that it was predicted by theory, and then people were like, oh my gosh, we can measure this. And so then the different standards, which are the carbonates grown at the known temperatures, those are made. And then we also can do things like produce CO2 gas that's equilibrated at a temperature. So we'll produce CO2 gas, and then we'll let it stand at 25 degrees C for a long time, and then we can run that through the mass spectrometer too. And we do that at a bunch of different temperatures.
Another question, how does this method compare to the Delta 018 record?
So that's a great question, because oxygen isotopes is kind of the gold standard for paleoclimate research. And so it is similar in some ways, being that they both look at the temperature relationships to heavy isotopes. And so they are similar in that way, but one major difference is that to utilize the oxygen isotope method, you have to have a known value of the oxygen isotopes of the solution that your mineral was made from. So say that you want to examine a coral for oxygen isotopes. You can look at the oxygen isotopes of the coral, but that value depends on the oxygen isotope value of the ocean water surrounding the coral. So a really neat thing about clumped isotopes is it does not depend on that. So you don't need to make any assumptions about what the solution value was. And so that was one of the most powerful advances of this method versus oxygen isotopes. You're welcome.
And Jamie, we have a couple of questions up on
Oh, sure.
from the Zoom audience. Maybe we can take, yeah, Steve Moore, the first question related, is it known or suspected why the Eocene period was so warm and why there was so much more CO2 then? And then Dennis asks, why were Eocene ocean temperatures so high? Greenhouse gases, why?
Okay. So let's see, is it known or suspected why the Eocene period was so warm, and or why was there so much more CO2? And I actually don't know why that there was so much more CO2. So there's probably people that could answer that for you, and I'm sorry, I don't know why there was so much more CO2 in the Eocene. I could tell you how that the CO2 value from the Eocene was determined, and that was using boron isotopes from, there's molecules in the ocean that are called borate, and they are very pH dependent. And so CO2 changes the pH of water, and so you can look at boron isotopes in samples similar to the ones we've discussed today and determine how much CO2 there would have been. So that's how that was determined, but why that occurred, I don't have a great reason for why. And it is believed, my interpretation is that a lot of the reason why the temperatures were so high was because the presence of a very high concentration. Oh. Oh my God.
Hi, Steve.
Thanks, husband. Oh, that's like gonna make me tear up, okay. So I think that it is believed that the high greenhouse gases in the Eocene was why the ocean temperatures were so high, because as we look back through the paleoclimate record, those two things are very tightly correlated. Okay.
We have a couple of questions on the other side.
So I'm curious if you can tell us a little bit about your next research project, what questions you're looking at and what methods you're using for that.
Absolutely. I actually just found out this week that I got funding to start a new research project that I'm pretty excited about. And yeah. And so the funding is for the next 4 years to look at, there's this suite of corals, and there are a bunch of different types of corals, and some of them are grown under controlled conditions, and some of them are wild-caught samples. And so what we're looking at is a couple different things. So corals produce these wonderful skeletons, and they're very heavily researched, but one thing is that it's not really fully understood how they make these skeletons, how they're biomineralizing. So kind of some of the more cutting edge applications of clumped isotopes is that these values, and like one of the other questions earlier, looking at more than one of these molecules that has clumped isotopes in it, you can measure more than one at once, and it can give you mechanistic information about the chemical reactions happening when a mineral is being produced. So we'll be looking at that, and corals are believed to make their skeletons from something called amorphous calcium carbonate. So it's kind of this phase that occurs before the actual crystalline skeleton. And so more and more research is being done of how corals and other biomineralizing organisms use this amorphous sort of phase to produce their skeletons. So we'll be looking at that. And then we also will be using them to look at paleoclimate research. So they're taken from a couple different ocean basins. So we'll be dating them and then using them to examine paleoclimate. So that's the next project. Thank you for asking that.
Hi, great presentation. It was so interesting.
Thank you.
I miss geology so much. I have a question. Was there any evidence, or that you know of or aware of, during the Eocene of mass die-offs of species in the oceans due to that temperature increases?
Oh my gosh, I should know that, and I am not sure. I'm sorry.
No, it's okay. I can look it up later.
I'm so chemistry heavy, and I feel like I lack a lot of the biology knowledge. I was just talking to Carol about that, but one of my goals over the next year is to like dramatically increase my bio knowledge of things like that.
We're literally just discussing that right now.
Oh, okay.
I just had a question about the method. You said that it was like around 2006 or so that it was developed?
Yes.
The isotope clumping? This caught my attention because it would have been actually 2006 or maybe 2005. Our geology class brought back some aragonite and calcite samples from Green River.
Okay.
From the travertine. And we had them go to the lab, and they put them in the mass spec and come out, oh yeah, it's aragonite or whatever. But what was the method that existed before then?
So
like how would they, because my memory's really foggy, but I think we were supposed to like figure out the temperature regime based on what came out of that or something.
Probably oxygen isotopes would be my
so
first
that's
guess.
what this gentleman was talking about.
That was kind of the most commonly used method by far for temperature reconstructions and it still is used, is oxygen isotopes.
Okay.
So that's basically a molecule that's very similar to what we were looking at, but instead of having the carbon-13 and oxygen-18, it would only have the oxygen-18. And so it would be carbon-12 with oxygen-18, and then the other oxygens would just be oxygen-16, so the lighter isotope. And that method is great and still used.
Were the machines very different?
No, they just weren't quite as sensitive. And so you can tune mass spectrometers to measure very specific masses, and depending on what's called mass resolution, you can measure molecules that have varying abundances. So the molecules for oxygen isotopes with just the one heavy isotope substitutions, those are in much higher abundance than something that has two heavy isotopes substituted in it. So these, the doubly heavy isotopes substituted, the clumped isotopes, like what we're looking at today, those are in, say, 1 per 100,000 sort of abundance. So you have to have an extremely sensitive instrument to measure it.
Thank you.
Mm-hmm.
Okay, well, one more. Dan, you get the mic this time.
I apologize for jumping the gun last time, everyone online.
That's okay.
Can you comment on the error bar of this technique, and what are the main factors that affect the accuracy?
Absolutely, that's such a good question. So the error bar depends on a couple different things. So it will depend on how many replicates you have. So that is a very key factor for any point that you see on one of our clumped isotope plots. There's a lot of replicates that go behind that. So we aim to have, it depends on what we're looking at, but a minimum of 4 replicates, meaning that we've taken the same sample, we've measured it at least 4 times and gotten good data at least 4 times before we will say that we feel comfortable, you know, that this is its value. And usually it's a lot more than 4 times that we measure it. So each time that we measure it, that error bar goes down. One thing also to consider is that natural samples typically have more variation in their isotopic values. So sometimes the error bar will be a little bit larger because your replication is not quite as good as it would be if you have, say, something that was produced in a lab. And so there's that. And so typically our error bars will equate to about plus or minus somewhere between 2 to 5 degrees Celsius. So it's pretty good.
Quick add on to that. Like, how far can you go back? And does the accuracy goes down? Does the accuracy decrease as you go farther back in time?
So there have been studies going pretty far back in time. So different things can happen in really old calcite or aragonite samples where you get things like recrystallization. So that's one thing that can happen where the crystal will no longer be in its original position. And so you'll sometimes get very kind of erroneous data from very, very old samples. You get things like partial dissolution. So you might be in some sort of environment where one of the isotopes has dissolved a little bit more than the other. So that can also skew your data. So one thing that people like to do to try and avoid situations like that is by using scanning electron microscopy to really closely examine the surface of a crystal if it's a very old sample. So one of the studies I showed here that was using the Eocene foraminifera, they had done scanning electron microscopy on all of their samples to make sure that there wasn't evidence of things like recrystallization.
So one of the major questions in the paleoclimate science is, do the temperatures and the greenhouse gases, sometimes one lags the other?
Yes.
And so there's the question of which causes which, because obviously there are feedback effects too.
Absolutely.
How do you, what are ways that you can help resolve that now? To what extent can we get, say, more accurate dating of
yeah, so
when
I mean,
things happen?
I can think of a couple different ways and ways that people are approaching that very problem. And so one way is to use multiple methods to look at the same thing. So say there have been some studies that have come out which use a totally different method that's also related to temperature, where you look at magnesium in carbonate minerals. And the amount of magnesium that you get substituted into say, a calcite crystal is directly related to temperature. So they'll reconstruct temperature with that. And then they'll use the same samples to reconstruct temperature using clumped isotopes. So if these two methods give you similar data, that is an indication that your data is good. And so that would be one way, is to examine that very issue, is using multiple methods, and also using the method of boron isotopes like I was talking about before, which is related to CO2. So if you couple all these different things together, it can help us get closer and closer to understanding that very important relationship of CO2 and temperature.